Okay, so now let's talk about the last part of our lab, the hidden Markov model and the TAD boundary identification. Okay, so we briefly talk about this HMM model in the lecture. That it's a statistical machine learning tool trying to help us identify hidden states from observation, from unobserved information through probability. Okay, so just a very quick recap, like what the hidden Markov model would be. Now imagine we just take the simplest example, coin flipping. Okay, I have a coin, I flip it for 100 times. I flip it for 10 times, let's say, and in 8 out of these 10 times, I got the head instead of a tail. It's not that unlikely, right? But we still feel it might be something wrong. So now it's more like from the probability perspective, how confident we are to call this coin to be a biased coin, to be a unstable, unfair coin. So we learned that from our statistic classes that we can use this Bayes rule to calculate the conditional probability. Now, what the Markov model is trying to solve, what the kind of question Markov model is trying to solve, is that now, imagine we don't have a very stable coin, as in the coin is either fair or unfair. This fairness is also becomes a random random event. Imagine now we have this coin, it has 50% probability to be fair and 50% probability to be unfair. But now if the coin is in the fair mode, then for the next event, for the next flip, the coin would be more likely to be a fair coin. Okay, so if you look at the diagram on the right, it's like if the coin is in a fair mode right now, next time it has 80% to stay in the fair mode. Similarly, if the coin is in the bias mode, next time it has 80% probability to stay in the bias mode. Now for a fair coin, you would have 50% each for heads or tail. But for a bias coin, you have a strong a preference over the tails than heads. So again, for this kind of system, we can define such a system. And the thing we can only observe would be the heads or tails. Now we have a sequence of heads or tails, say 100 flipping samples. We have a sequence of them, different patterns and different percentage. Now this time, how confident we are to say this coin is stable or unstable, or to say what is the probability behind the different hidden states. Okay, so this is what kind of hidden Markov model is trying to solve. The reason it's called hidden Markov model. First of all, because one, the state is hidden. We don't observe directly the states. And two, the system is Markovian. Markovian's just a very quick recap means, the probability of one event only depends on the previous event happening right before it. So that is, you only depends on the last minute, and your last minute only depends on the second last minute. So that is what we call a Markovian. So here in this state, this time, when we flip a coin, when we are imagining this hidden state, we know now this time, I flip it. Now for this particular point, for this at this particular time, I have a fair coin. So next time when I flip it, like the state of the hidden state of that coin would only depends on the hidden state right before this event. I wouldn't care that much about what's happening before 5 minutes ago, because I only look at the last hidden state. From our biological question perspective, why this is interesting? Because now imagine we are looking at a chromosome. Okay, we're using all this Hi-C, all these fancy techniques. We observe a lot of genomic interactions events. Now we know that if two regions are closer to each other, it is more likely to get a cross-link event. But we also know that due to the randomness, the stochasticity of the cellular system, of the nucleus, is also fairly likely to get a cross-link just from a random interaction. Now the data we get from Hi-C would be a sequence of events, a sequence of interactions. We know that some of the interactions are because they are spatially closer to each other. Some of them they are not. But we also know that if we see a cluster of interactions, right? If we have a specific region of the chromosome of the genome where we see a lot of interactions, within that region, then we know they are probably interacting with each other, or spatially co-localizing to each other. So this is where the hidden Markov model can really help, because we do not know whether these regions are interacting or not, actually, because we just do not have this ground truth, and we would never have this ground truth, because the only observation we can make would be whether do we have this interaction event or not, instead of whether we are spatially closer to each other or not. Okay, so with this all set up, we can say, we can use all this, developed hidden Markov model that has been published out there. You have multiple different versions everywhere else. We will use the one we talk about during lecture, and also in the homework we also attach the link to the template of the code, to the algorithm. Okay, so now we imagine we have this system that has two states, up or down. Okay, we just artificially define it. We say the initial state for this system. So first of all, in total, you only have two states, two hidden states. But imagine now we have 8 observation states. Let's come back to this 8 later on. Let's just continue on this hidden Markov thing. Now, I say initially I have 50-50% probability to be in either state A or state B. Okay. From the transition probability between state A and state B would be 90% or 10 versus 10%. So we have 90% probability to stay for one state, stay in one state, and we have 10% probability to flip sides. And we can also define a emission probability, like we talk about, like the observation probability. Now imagine we are in the state 1. What would be the probability of the observations we get? How frequent that would be? Or if we in state B, I'm in more immune mode, and I do not see a lot of interactions. But still, we have a probability distribution of how many events I would observe. Okay, so with all this setup, this now becomes a hidden Markov system, and we have a data, and then we can try to use the data to fit this model to see if this model is correct or not. Or the other way around, we can define this system first, and then we can make a sample. We can draw a sample. We can draw a sequence of samples, a sequence of events, based on our definition of the system. Now you may wonder, where are we getting this 8? So let's go back to this slide. So now the data we get from Hi-C is a continuous. You can consider that as a continuous observation, because the interaction can be 0, it can be 1, can be 10,000, can be 1 million. Even though theoretically speaking, they are still discrete. But if you imagine each single observation, each single value to be a separate state, then your system becomes too large. There's no realistic data set can be used to solve this problem. So now, as a compromise, we're trying to do the binning like we did in the previous questions. We're trying to bin those continuous distribution into different separate regions. And in R, we are using this function called cut. Okay, imagine we have this just in case some of you haven't used this cut before. Imagine you have this X, you can generate this random uniform array where you generate from 0 to 1, you draw 100 samples. And now you can use this function cut to say, I want to cut this into 10 different bins. So it would generate a categorical array saying which class would your sample belong to. So instead of a continuous scalar value, now you have a categoric label of which region, which bin region you belong to. Similarly, just like similar to the histogram, you can also define directly the thresholds for the binning. You can define the number of bins you need, or you can instead you can define the coordinates for different bins. Now, so we load the data. We load the data, and it's something like let me see if I have a figure here. Give me 1 second. No. Sorry, where's my R? No. Okay, you cannot see it anymore. Let me share. Let me share the full screen. Okay, so imagine this is the data we have. Okay, we have this data. We provided you the data from the homework. Here I'm just using this data, so try not to copy the results. Just try to develop your own version. Now, if I load the data, and let's now look into the data, what the data looks like. So similarly, we have the region, the genomic region. So chromosome first, and then start point, end point. And we have this value, it's called DI, it's a direction index. In terms, in regarding to how do we define this DI, you can refer to the link we also attached in the homework. But just imagine this is something we want to predict, or we want to bin on. Okay, so this is kind of the data where for each bin here we have a value. For each bin here we have a value. So now imagine if we calculate just a middle point, like a midpoint between end point and start point. So just trying to merge these two columns to sorry, I cannot click it anymore. It's the zoom is hiding everything. Okay, so now imagine I'm just trying to merge the column 2 and column 3 to one column, so that's easier for the following up questions. Because as you can see, the bin size is relatively small comparing to the actual value of the region. So it's only 40,000 comparing to 5 million. So we can just ignore the difference. But we just call this to be the label of this class. And now we take this middle point and we calculate it. And now we plot the data. The x is the midpoint. Sorry, the y is the midpoint, and the x is the DI score. Well, actually, that's incorrect. So x is the midpoint, and the y is the DI score. Okay, so now imagine actually now what we are looking at is a chromosome, right? It's a chromosome where for different regions you have a different DI score. As you can see, you have some ups and downs. It seems not that random because you see some clusters. For example, here you have this specific region where you have much more frequent, ups than downs. Similarly, also here you have a lot of downs than ups. So our job here is trying to train a model that can separate this data into the two hidden states. You might argue, I just draw a line here from just at the y equals to 0, and all the points above 0 would be, I would call it an up, or all the values below 0, I would call this a down. So the artifact of that, the pitfall would be you are ignoring the stochasticity of the system. So you might end up with a lot of false positives. And also, you are undercutting your power because you already know with your prior knowledge that a cluster of events would means more information. So if you just draw a line here, you are basically making each prediction for each dot to be an independent prediction. So you are underpowering your data. So that is kind of the rationale why we are using this hidden Markov model. So in the end, what we want to have is something like do I have the figure? I do not. Let me try to run it. So just very quickly to get you a sense of what we are trying to predict. Okay. So this is what we want to do, right? It's the same figure from this one, but now this time we assign a color for downs or ups. As you can see, for those clusters, for those big clusters, we would be able to manually pick it. But for some of the small ones, for example, here, if you can see it clearly, some of the events, they are actually, even though they are above the x-axis, we are still calling it to be a down state or to be an up state. That is what the hidden Markov model can give us. Okay, so go back to our slides. So we are defining our system in this way, and we are using the code from you can use the code either from our lecture, or it's easier maybe to use the template from this link. We also attach it in the homework. So what this looks like is, this is what the block we put it there, and you can just go down for the here I'll talking about examples and the theories behind the hidden Markov model. So you are all recommended to read through it. And they have two versions. They have a Python version. They also have R version script. So it's fairly easy. You just adapt this function and trying to put in your different inputs. So here, what is your transition matrix? What is your emission matrix? What is your initial probability distribution? Okay, so with all this, you should be able to directly calculate a figure like what we are having like here in the slides. And trying to just do a manual check, just do a sanity check after you train the hidden Markov model to see if their prediction is reasonable. So if you see a mosaic prediction here, even though for some of the regions you can directly tell that should belong to one state, but instead the model is giving you an unreasonable prediction. Try to debug the code or try to change your training parameters, for example, number of iterations, etc. Okay, so that will be our homework 3, and that I would actually conclude our lab, because of part 4 is more about just question and answers. There will be no, it's more like conceptual. There's no coding, not that heavy coding required for part 4. So we wouldn't cover that too much in the lab. But still, you're more than welcome if you have any unclear point you want us to clarify. But we wouldn't help you to solve this to answer those questions. Okay. So I just a conclusion. I know this 2 weeks can be quite overwhelming. Because some of the problems that we are developing are actually, this year is actually our first year to trying to do this Hi-C and something related to this chromatin genomics. So if you feel sometimes it's a little bit lost, a little bit confusing, you are not alone. Okay, you're not alone. So please feel free to reach out to us if you find anything unclear, and try to stick onto this lab slides, trying to stick onto the instructions step by step, so that you can with the how to say, it's more optimistic, to stay on the good track, right? It's more hopeful to stay on good track. Okay. So with that, I will stop the recording now, and I'll see you guys on Thursday, Friday, and Saturday. All right. See you later.